Imagine a world built entirely from just 20 basic blocks. I mean, one amino acid, 20 types, but infinite structures. Welcome to the literal foundation of biology.
Welcome to this BioInfoBytes explainer. Today, we're taking a rapid-fire journey zooming out from microscopic linear sequences all the way up to the three-dimensional machines that keep us alive. We're talking protein structure, primary to quaternary. Let's get right into it.
So let's dive into this master blueprint of biological complexity. Our anchor for today is human hemoglobin. As you can see, we start with the beta-globin chain, just a simple linear sequence over on the left, and we're going to watch it continuously fold until it becomes that totally intricate molecular machine on the right.
Starting off with primary structure, the linear sequence. Think of this level as the raw sequence. It's literally just a linear polypeptide chain of amino acids assembled directly from DNA instructions right on the ribosome. They're strung together like beads on a string, locked tight by peptide bonds. And this base sequence, it absolutely dictates everything else that follows. Moving on to secondary structure, local folds.
That straight line we just talked about, it immediately starts interacting with itself, folding up into these really specific geometric patterns. We get coiled, rigid alpha helices and flattened beta sheets. And both of these architectures are beautifully stabilized by a continuous network of hydrogen bonds. And when you look up close, you can clearly see these tightly coiled cylinders locking right into place. This is essentially the sturdy structural scaffolding of our entire protein.
Next up is tertiary structure, 3D conformation.
At this point, the protein folds out into actual three-dimensional space, driven by a pretty potent cocktail of chemistry. We've got a hydrophobic core actively hiding from cellular fluid, locked together by disulfide bridges, ionic bonds, and van der Waals interactions, packing the whole thing into a highly specific shape. And here's that exact theory in action. The human beta-globin chain perfectly folds its helices into this incredibly specific, compact globular domain. Now, it looks ready to work, right? But for a lot of proteins, this actually isn't their final form.
Which brings us to quaternary structure, subunit assembly.
Because here's the thing, many proteins just can't function all by themselves. So here is our hero moment, hemoglobin. We have 4 individual folded chains, 2 alpha and 2 beta, assembling non-covalently into a massive functional tetramer, complete with 4 little heme groups perfectly nestled inside to carry your oxygen. Now, why bother building this massive complex at all? Well, it enables something called cooperative binding. When just one subunit grabs onto an oxygen molecule, it physically changes the shape of the entire complex, instantly making it way easier for the other 3 to grab their oxygen too. It's honestly just pure biological efficiency.
Let's look at applications. Predicting protein function.
To predict disease and understand these structures, scientists follow a really strict pipeline. First, you select a target and amplify the coding sequence. Then, you determine the crystallization or NMR conditions. After that, the real magic happens computationally. We use algorithmic tools, like the Pol server or 3D PSM, to actually calculate these complex 3D models. Because in this field, knowing the exact shape completely unlocks the function. But I want you to consider a really critical question here. What happens if just one single amino acid in that initial primary sequence mutates to a valine? We're talking about just one tiny bead changing on this massive string. Well, the answer is pretty profound. That single linear error cascades all the way up the structural ladder. It changes the local folds, which then alters the 3D tertiary structure, and ultimately physically warps the entire hemoglobin complex. The end result of that one tiny change? Sickle cell disease. So it all comes down to one absolute, inescapable truth that I want you to take away from this explainer. Structure determines function. If you change the primary sequence, you change the entire structure, and you literally alter the biological reality of the organism. As we keep building better and better predictive algorithms, this really remains the ultimate puzzle, decoding the incredible, complex origami of life.